I wanted to make a brief video to talk about the concept of survival analysis and some of the, theoretical background of survival analysis to make it a little bit easier to actually understand how it's working when we do it in R. So survival analysis is one name given to a kind of analytical approach that is in general dealing with data where there's a certain time until an event happens. And typically it doesn't have to be this way, but typically that event happens and it happens once per individual. So survival is a kind of an obvious example of when an event happens just once per individual. I'll talk about other kinds of time to event data, but for now, let's just think about it in terms of like how long an individual survives for. These data are usually a continuous response, so think about it in terms of like the number of years that an individual lives. These data are frequently right skewed, so of course that means that the mean and the median are relatively low compared to the extreme values of the data. And so there's a really long tail going off to the far right. These data are not normally distributed, usually can't be made to be normally distributed. And so you have to deal with them in a slightly different way than we would normally be used to in terms of like a t-test or an ANOVA or something like that. These data are also censored, and I'll talk about what censoring means, but this is a real important component to the process of doing survival analysis. And there are lots of medical examples that you'll run into. Time until a person dies, so strictly speaking, survival analysis. It could also be time to the onset of a particular disease, like how long until a person comes down with X disease, or how long until that person relapses. So they might be cured of cancer, and then that cancer might come back after a certain period of remission. Could be the length of stay in a hospital. So you stay in a hospital for a certain period of time, and then at some point you leave the hospital. So there are a lot of medical examples. And for that reason, I think it's real important for students of biology to be exposed to this concept of survival analysis because there are just so many applications in medical fields. But despite that, there's lots of other applications of survival analysis beyond medical fields, so just general biological applications. So how long does it take until an individual plant flowers, until its flowers open up? How long does it take before an adult insect emerges from overwintering or from the pupal stage? How long does it take for a seed to germinate? All of these things happen typically once per individual. So a seed will take a certain time, and then at that certain time, the seed germinates. So we could be interested in the time until that seed emerges. There's other examples, birds migrating, eggs hatching, etc. So lots and lots of different kinds of biological examples. This is an example plot of post-operative survival of individuals. So this shows the right skewing of data. This is a particularly risky surgical procedure. Many people do not live very long past this surgery, but you can see that there are some people way out here on the far right where there's a really long persistence time. They survive a long time after that survival. So after that surgery. So if we're interested in long time periods until an event happens, and there is this strong right skew of data, again, traditional normality-based statistical processes or procedures are not going to be appropriate here. The other major thing that highlights the need for survival analysis as a separate kind of data analytic technique is this issue of censoring. So right censoring is the one that's going to be the most common. There are other forms of censoring. Real briefly, survival analyses is kind of an inherent part of a survival analysis. You have to deal with censoring. Censoring occurs when we have some information about an individual's survival time or time till event, but we don't know the survival time exactly of those individuals. So censoring can come about in a couple different ways, but like I said, right censoring is the more common one. And so imagine this example here where we are looking at an individual, and the individual we know, let's say, was born on this time, and then they live until this time. So we know that time until this event has happened to the individual. What about an individual over here that started, off to the left of this time period? They went on, they went on, and they went on. And at a certain point, we're just done with the study. We stopped the study at this dashed vertical green line, and we don't know when they died. So sometime after the study ended, but they were alive at the time of the end of the study. Similarly, we've got another individual here that entered the study fairly late, but then again, we sort of stopped the study at this time point. We don't know at what point the event happened to them, but we know that they made it until this time. So censoring allows us to take into account this ability for an individual to persist until a certain time, and then after that point in time, we don't know what happened to them. Left censoring is the same kind of idea, but we don't know when they entered the study. And interval censoring allows multiple time points along the way where someone can sort of leave the study or leave our monitoring of them. But we're not going to get into that. Mostly we're just going to be dealing with right censoring. The data formatting for a survival analysis is a little bit different than we might see for other kinds of analytical approaches. So this first column will be the unique identifier. So this will be, say, the observation of those individuals. The next column is maybe some kind of treatment or categorical variable. In this particular case, we have controls versus mosquitoes exposed to permethrin. And we've got, any number of other categorical variables that we can look at. In this case, collection site, history that they've been exposed to, either spray or no spray. And then, so here we get to the things that are a little more unique with survival analysis, because survival analysis is about time until an event happens. We have to have for our main response, it's the time unit until the event has occurred. So here we've got, it was 120 minutes until the event happened to observation number one. 120 minutes until the observation happened to individual number 12, and it was only 30 minutes until something happened to individual 26. So sample number 26. So we've got our time until event column, and then we also have this censor column. The censoring column usually is a binary, so either 0 or 1. And usually you have to tell whatever program you're using what the censored value is. So we can tell from the layout of this particular data that 0 in this case means censored. They lived through the end of the study period, which in this case was 120 minutes. And we didn't know what happened to them, how long it took for them to die. This was mosquitoes. So how long did those mosquitoes live until they died? Well, they lived 120 minutes. We were done with the study. We didn't know what happened to them afterwards, so we censored that observation. We know they lived at least 120 minutes. Whereas the individuals down here, they were they were not censored, so their censored value is 1. And we know that that individual died at 30 minutes or at 45 minutes or 60 minutes. So the censor column is real important. We either know they lived until a certain period and then our study was over, or we know that they lived until a certain period and we actually observed the event happening to them. So you've seen kind of the distribution of what those survival times might look like. We're not going to get into too much of the math about survival functions, but basically we're trying to estimate the shape of a survival curve. If we start off with 100% of our individuals surviving at the beginning of a study, and what's going to happen is as time goes on, fewer and fewer of those individuals are still around, so we are starting to lose some individuals as we move down this curve. As we go down this curve, eventually our study is done. We stop the study at some time period. Now this has time going off into infinity, but we could stop the study at any time. We've got this survivorship function that's happening theoretically as more and more individuals are sort of passing from one state to another. In practice, what survival analysis tries to do is it tries to estimate the shape of that survival curve for a particular population. So this population may be the individual insects collected from a particular site or exposed to a particular treatment, or seeds that were germinated in one category or condition versus another. So this is an estimation of one single survivorship curve. And everything that happens whenever we get these jumps down indicate that individuals are surviving, so the event is not happening through here, and then at some time period we record a bunch of individuals. The event happens to them, and then for another sampling period we get no events happening, and then all of a sudden a bunch of events happen. When you see a stair-step pattern like this is usually when the observer is looking, sorry for these horizontal lines here, but at these points, this is usually a point where the observer, the experimentalist, is sampling individuals. So we don't necessarily know what happened in the meantime, but when we check in on them again at this time period right here, we see that a whole bunch of them have died. They have no longer survived. The event has occurred to them. There's several statistical approaches for the ways that this survival curve function is estimated. One of the simplest ones is known as the Kaplan-Meier estimate. This is a non-parametric estimate, so we're not making any assumptions about normality or any aspects of the parameter or the parametric distribution of the survival of these individuals. It's very commonly used to describe survivorship, so it's a real frequently used approach. It's a good way to compare two study populations at a time. So it's kind of like the survival analysis version of a t-test, essentially. So we're just comparing two separate groups. It has a very intuitive graphical interpretation and representation. There are lots of others. The most commonly used alternative approach is this approach known as the Cox proportional hazards model. We're not going to talk about that. We're not going to use that. It allows much more nuance, and it's a much more powerful technique in some ways. It's a more flexible technique, I guess I should say. So here are a couple examples of applying survivorship curves or survival functions to different populations. So here we're looking at the survival of all individuals, humans, due to all causes of mortality in New Zealand and Australia versus, say, a country in Africa, Chad. So you can see that New Zealand and Australia are very close in terms of survivorship functions and survival tables, survival figures, whereas the people in Africa have a much faster death rate or a higher death rate. In a Kaplan-Meier plot, you can see we get kind of these, one that's actually estimated from real data. You can see that we get these steps. Again, these steps right here represent times when the observers have recorded individuals doing different things. Are they still alive? Has the event happened to them? So this is the estimator of the function right here, and then we can get confidence intervals that go around that estimated curve. Some of the summary statistics that we get from doing a survival analysis, because this is non-parametric, so we're not making any assumptions about a normal distribution of these survival times. We're not going to look at mean as much. Instead, we'll typically compare medians, so a time at which half of the individuals have had the event happen to them. This does require a fairly substantial amount of data to get a good representation of the distribution by looking at medians. You can also pick you can look at mean. You can also pick a particular time period here. We've just kind of mentioned 5-year survival, but you could pick 5-day or whatever. You could say, at this time of my study, there was this kind of difference between my two groups. So there are lots of different kinds of summary statistics that you can look at and compare. The Kaplan-Meier and life table approaches, so that's another alternative. They're both non-parametric methods, so no assumptions are made at all about the distribution of the survival times. The typical distributions that we see in survival data are exponential and Weibull distributions. You can apply these to a Cox proportional hazards model. They tend to be more powerful, but you have to really know what you're doing with the estimation of the distribution of those survival times. And so they can be, although more powerful, they can be, they're more of an advanced approach. And since we're just talking about an introduction here, we're going to stick with a Kaplan-Meier version. Here are just, though, some examples of different kinds of distributions that you might get based on a variety of different scenarios. So again, if you were using a proportional hazards model and you were using a specified distribution, then these distributions are going to vary across the different kinds of scenarios that apply to your example. So we're going to be interested in comparing two groups. We're going to use Kaplan-Meier survival analysis. And so what you might get, for example, if we're going to compare the median, again, we're going to compare the median at this particular spot. So we're going to look at how long did it take group 2 to reach this 50th percentile surviving, and then how long did it take group 1 to reach this 50th percentile surviving. And so we can compare these median values, and here the median is 8 for the placebo group, and the median for the medicinal treatment group is 23, so we've had some kind of intervention. What we would report then is, look, the median time was nearly 3 times longer than the placebo group that did not receive any intervention. So that would be the kind of thing, the kind of statistic that we would give or report from a survival analysis. Similar kinds of survivorship curves or survival analysis curves for comparing immunocompromised individuals versus people who were not immunocompromised. The visual layout of these kinds of figures is pretty straightforward, right? You can see that individuals that are immunocompromised tend to die very quickly, so they have a very steep survival curve, whereas people who are not immunocompromised have a relatively shallow survival curve. And so again, we're talking about the intuitive nature of these two group comparisons using Kaplan-Meier survival. You can really see that in these kinds of figures. The statistical table that we would end up getting is both a mean and a median of the two groups. One shortcoming of using the median is, notice that in the non-immunocompromised individuals, there is no estimate for a median, because if we go up here, if this is the 50th percentile, these individuals never actually cross that median. So sometimes median is a good statistics to report, sometimes it's not. So you might have to go with the estimated mean here versus here. Ultimately, the statistical comparison that generates a p-value, if we're comparing two groups, is a log rank test. In this particular version of the log rank test is a Mantel-Cox test. We get a chi-square value. If we're comparing two groups, then we get 1 degree of freedom, and then this is our p-value. Whoa, that went all crazy. Remember that even though the program, whatever we're using, R or SPSS, might not give us the exact p-value, there is no such thing as a p-value of 0.000. So we would report this as less than 0.001, for example. Here are a couple of ecological examples. So instead of counting off individuals who have died, we can look at the accumulation of individuals. So this is the accumulation of individuals caught in a moth trap. This is a species of moth known as Desmia funeralis. And we're looking at 3 different sites. You can see that they get captured at a faster rate in this location, a kind of intermediate rate in this location, and the slowest rate in this location. And if we compare the median values, so the 50th percentile would be about here, and you can see that 50th percentile occurs at around this number of days for the green group, and it takes this much longer for the red group to accumulate that many individuals. The other thing that I was looking at here is at how long does it take individuals to, well, we're going to skip that part for now because it's not really helpful to our general discussion. You can also think about accumulation of seeds germinating in different treatments. So we've got cumulative proportion of germinating seeds. Just like with the moth example before, everybody or all your 3 populations or 4 populations start off at 0, and then they start to accumulate some numbers over time. These are very smoothed examples because in this particular experiment, I had thousands of individuals in each treatment. And again, back to that intuitive nature of these figures, you can just kind of see the slower accumulation in the high salinity treatment and the very rapid accumulation in the 2 lowest salinity treatments. You can see that basically the interpretation is seeds germinated a lot higher in low salinity and germinated a lot slower in high salinity. So interpretation is really quite straightforward. That's a brief introduction to survival analysis, so the concepts behind it. And what we'll do in class is we'll run through an example on how to code this. I hope this was helpful.